Well, thanks very much. Thanks very much for the kind introduction and invitation. And I'm happy to tell you a bit about open microscopy in the life sciences, the topic sharing is caring. So what my group is interested in, let's see, cursor here, is how life and in general also food organizes at the subcellular and microstructure. Yeah, so what we see here on the first image is a kind of very simple bright field imaging of single bacterial strains. Yeah, so they're a couple of micrometer long, and they all look in the very beginning very unassuming. Yeah. If we now zoom in a bit, then we starting to get a very complicated picture, and I switch on my laser pointer, where a lot of research has basically determined the structure of many of those proteins in the cell. Yeah, compared to the space, if you look in the sky, yeah, the space is fully cramped. Yeah, so there's basically no space at all available. You have very tiny proteins in the nanometer scale. They're interacting with each other. They're moving around. They are dynamic. Yeah, and many conventional techniques like X-ray crystallography or so, they are not able to cover those dynamic aspects at all. And the big breakthrough in the entire life science imaging field was basically the discovery of the green fluorescent protein, which then for the very first time allowed you to tag any kind of gene of interest and look at the position and the dynamics of certain proteins. And the issue now people had is that physics comes into play, and rather than seeing that single protein doing something, what you see is a big blob. Yeah, so that's what we call diffraction limit, and that limits our resolution, conventional microscopy, to around 250 nanometer. And how do we go back to that? And just look for the concept to resolve those two structures. Let's say you have resolution limited to 250 nanometer. You have a structure that has a feature size smaller than 250 nanometer. And one possibility is that you find conditions in which most fluorophores are in a dark state. So they cannot actively image. They are not fluorescents. If you now find conditions where a subset of those molecules blink, for example, then you are starting to be able to localize single emitters. And because we know the shape of the emission, we can apply, for example, a two-dimensional Gaussian to fit the position of those molecules with a rather high precision and accuracy. If we continue to do that for many different frames, yeah, we can resolve the underlying structure. And basically what we do, at least in the single molecule localization microscopy field, we start to separate those emitters in time to separate them in space. So what I now explained to you is kind of how to conceptually resolve small structures. But the question is, how do we practically resolve those structures? And what we need to do that is a combination of good hardware, good software, and suitable assays. And all of them are kind of equally important. Yeah. And the challenge we as researchers face, that we might not have the expertise to do that. We might miss the funds. And for me personally, time and money always goes closely together. Yeah. Sometimes you would like to buy, but it's always a question of time or money. Yeah, things get cheaper if you have unlimited amount of time, but normally you neither have time nor money. Okay, so how can we address that? And that's what we're here for. Yeah, so we think that with open microscopy, you have a very good chance to addressing all of those needs and challenges. And this is still a relatively defined setting, yeah, because I'm talking from the academic perspective. I have, let's say, a biological problem that I want to solve, and I'm interested in microscopy. So in a way, I'm still kind of in this rather tiny fraction of a closed research environment. Yeah, what I'm still not looking at all is, okay, from the society perspective, is there any need to do my research? Yeah, how do I use my primarily public funding to do that? Can people access it from the outside? Do they have a chance to see what I'm doing with the money? Yeah, and so there's a whole thing outside the closed research environment, like sharing data, collecting ideas, discussing ideas, that is often, yeah, not even thought of. Yeah, so together with a couple of colleagues, some of them who will talk later here in the session, we kind of put all our thoughts together and thought a bit about open microscopy in the life sciences, what is needed, and where do we think it's going. So if you're interested in the topic, please have a read, and I will pick only a few of those aspects which we discussed. So the open microscopy, first of all, we had to kind of find a definition. Yeah, so what do we mean by that? And we said, okay, we define it as a movement to make any kind of scientific research involving microscopy, associated data, in dissemination of accessible to an inquiring society. Yeah, so we cannot define who the society is. Yeah, that could be the taxpayer, that could be us as researchers. Yeah, and what is associated data? Yeah, so this includes, for example, hardware aspects, so how to build something, how to maintain it. It includes the assays, so what to measure and how to measure. And of course, equally important, it also includes the software. So how do I analyze my data? And for software, this has always been a bit easier, because people just uploaded their software packages on the internet. And for hardware, this was kind of a bit still open. So what we came up now already almost 5 years ago is we developed the microscopy platform with the idea to make kind of super resolution microscopy easily or easier accessible. Yeah, so people here, Sander Baas, Koen Martens. So basically what we did is we developed a couple of parts, so parts of them 3D printed, parts of them milled in a workshop that you can set together, relatively basic super resolution microscopy, building, of course, on many different works. And our idea was to kind of limit the degree of freedom. Yeah, so we didn't want to do something which can do everything, but we want to narrow it down so that it can do, for example, application super resolution microscopy easily and to a good extent. Yeah, just to provide you of the updates, a couple of things we did recently. Yeah, so in this entire microscopy platform, yeah, so there you see excitation part, stage part, objective, and so on, and camera. So we added 2 editions in a recent publication. Yeah, so one of them is kind of a flat field illumination. Yeah, so where we use an optical element that transforms your Gaussian intensity laser intensity distribution into something which resembles very nicely over almost more than 100 square meter a flat field, yeah, which simplifies, gives you better heterogeneity in the sample. And then to the right, into the detection path, we implemented deformable mirror that allows you, as in kind of adaptive optics, for example, if you want to do single molecule measurements in turbid samples, that you can correct a bit for the aberrations. So what you can see here is that this point spec function in our defocus, they always have kind of a tilt into a certain direction. And by improving over all those modes, fitting that accordingly, you end up with things you would like to end up, and that is in most simple cases very symmetric patterns of the point spec function. So here we used, for example, then we combined kind of a hardware implementation with software implementation from the Lucas Kapitein group in Utrecht, which already did this for a different deformable mirror, kind of did this software correction. Yeah, so again, without them disclosing their information, making it openly available, we would not have a chance to implement this in reasonably amount of time. So from the MyCube, which luckily a few people built, there are now more iterations going on. Yeah, so I would like to point out 2. Yeah, so one is the so-called MyEye from the Tutkus lab in Lithuania, who started with the MyCube and then thought, we can do a few things better, and we can replace even more of the components that we kind of bought commercially with more homemade equipment. So for example, using laser diodes rather than a commercial laser box, or using kind of really cheap cameras rather than the scientific grade cameras that we used. More importantly, they included a proper infrared autofocus path. So if you're interested in that, then so this was recently published and is already available online in HardwareX. And then I'm looking forward to hear later from Paul French about their concept of the open frame microscope, which without saying too much about it. Here, interesting aspect is that you can actually start buying some of those comments from a commercial vendor. Yeah, so you don't have to go to your workshop, but that increases also the accessibility by just pressing somewhere a button and order certain components. So now say we have my group, we have my group, an open frame, and then I like to bring up this XKCD comic about standards. Yeah, so there are 4 different standards. That's ridiculous, way too many. We should have one that covers everything, yeah, until you run into a situation where you have 15 competing standards. Yeah, and in the past, I always saw that a bit like as a problem, but nowadays I got way more relaxed about that and saying it is simply the way it is. Yeah, it's good if there are different solutions for different requirements from different people. Yeah. Then in our publication, we came up with kind of very rough guidelines, 7 guidelines, which I will just briefly mention here. And those are things that you can think about if you develop or think about developing an open microscopy platform or small aspect of it. Yeah, so first question is uniqueness. Are you inventing something new? Are you iterating on something? Would it, for example, could make sense to contribute to a different project instead? Yeah, do you have to do you want to establish a new standard, or do you say, okay, maybe I want to contribute to something other? Then if you open it up, yeah, so what are the resources? Can you as a group, can you sustain the development for longer? Yeah, and that's always a very important question, because you might always have a very enthusiastic PhD student who then eventually graduates, and then what happens then to the project? So then again, do you want to involve others? Yeah, do you want to build a community around that, or do you say, okay, I don't really care what's happening to that? Then I think very important, 4th point is documentation. I think the rule of thumb is document as carefully as you possibly can, because there is always something which will be vague and other people might wondering about later. It's very good if you keep a certain interoperability. Yeah, best is of course if devices and solutions can be combined in one way or another. First, what is the need? Yeah, so who needs what? Is there, for example, a large community like in the selective plane illumination, spin microscopy, where there, for example, were no commercial solutions available at that point, where people really had to work together to set something up which works for many? And then can you also use open microscopy as a way of merging expertise from different groups and people? Yeah, again, expertise could be one lab in the software, another group in the hardware. If you combine everything, then you end up at more than the sum of the parts. Yeah, something I would like to put your attention in there. So we kind of, with all the authors, we came up with a repository on open microscopy. So it's a GitHub page where you can find long lists of open microscopy projects and people involved. So please have a look there. If you think you're missing a project, or if you would like to have your project on that list, then please send us an email or just pull a request on GitHub to get added. And now let's change gears a bit. We want to talk about something that we recently published on spectrally resolved single molecule localization microscopy. And I kind of want to link that a bit to the idea of how we understand open microscopy and open hardware. Again, as probably many people already do, manuscript went to the preprint server so that people can have a look, get acquainted with the idea, ask, for example, questions already or provide comments, and then still kind of the traditional way we go for a journal to get it published and get the peer review going. So what's the idea there? Here I show you the detailing of the emission spectra of green fluorescent protein and 2 dyes. And as long as you are here, they are relatively wide separated. Yeah, you can select certain windows and say, okay, I detect here bluish green or orange or red fluorescence, and that's all fine and good. You run into more issues if you want to, for example, say, okay, I don't want to excite in the blue because then I have too much background in my cells. I want to excite only in the red. Yeah, if you're then trying to select matching fluorophores, you will face the issue that the emission spectrum of all those fluorophores closely overlap. So what can you do about that? And the idea we had was, again, building off many different manuscripts, to take a transmission grating, which is placed, so it has kind of an angle, which splits the incoming light into a 0th order and a +1 first order. Yeah, so basically in the center, you will see what we see as a point spread function, now a single molecule localization microscopy. And next to it, we have kind of an elongated point spread function where the distance between those 2 points tells us something about the mean spectral emission of the fluorophore. Yeah, so the main idea is now to bring that transmission grating as close as possible to the camera chip, yeah, or to the image plane. That way, we minimize the distance, we minimize the dispersion, and we can approximate basically the 0th and the first order by a single point. Yeah, this allows us then to take standard super resolution localization algorithms to determine the center of mass or the center position of every fluorophore. How does it look in the simulation? Yeah, so we have first order is shown here, and then a couple of micrometer next to it, depending on the characteristics of the grating and the distance. You will have another dot, slightly elongated. By then checking the distance between those 2 dots, we can determine the center emission wavelength of the fluorophore. How does it look like in the experimental data? So this is here a couple of single fluorophores, and you see single emissions, and you always see this pair-like features distributed all over this over the screen. And to give this now a go, yeah, what do you need? Yeah, so simple. You buy a diffraction grating, sets you back around 120 euros. Then you use the software. So we developed a fast temporal median filter ImageJ-Fiji, which is many people use for image analysis. And ImageJ-Fiji, you take your favorite super resolution localization algorithm. So for example, Thunderstorm. And then you download the software that we released with that to do a very fast pair finding analysis in ImageJ. Yeah, so you first localize as normally all your molecules, then you run this to get the distances. Yes, you raised your hand.
This is your 1 minute warning.
That's my 1 minute warning.
Yes.
That's very good, very close. Okay, so how does it look in reality? Yeah, so here we did some storm images where we labeled with 2 different fluorophores, and you can see the pseudo color coded pictures. So can we nicely resolve different structures, the microtubules in the clathrin pits, and then by selecting areas with summing up only the green fluorescence or the red fluorescence, you can nicely now separate even largely overlapping fluorophores with their spectral features. And the last data slide is then we applied this for DNA paint, where we simultaneously excited in the green and the red, but we used 3 different strands labeled with either a green dye or 2 different red dyes. And then we came from this diffraction limited latex beads in the DNA paint approach to do the super resolved latex beads, which are now also distinguishable in the colors. And with that, I'm coming to my last slide. So I hope I'll convince you that open microscopy aims at improving the accessibility of high performance microscopy. Yeah, I presented a couple of options for improvements, so adaptive optics, spectral resolved SMLM, and very importantly, your contribution matters. Yeah, so whoever is working on a project, please feel encouraged to open up on what you are doing, get in touch with others, and start sharing your work. And I would also like to thank specifically the team on the open microscopy paper, Benedikt, Barbora, Emanuel, Seamus, Wiebke, Robert, Hase, Kurti, and you for your attention. Thank you.